Good afternoon and good morning, everyone. Welcome to our webinar on MetaSV, an accurate and integrated structural variant caller for next-generation sequencing data. My name is Anoop Grewal, and I'm Product Marketing Manager at Bina Technologies, and I'll be co-hosting today's presentation along with our Senior Bioinformatics Scientist, Margoob Mohideen. I'll start with an introduction, but before that, let me remind you that we'll have a question and answer session at the end of the presentation. And you'll notice there's a green bar at the top of your screen. If you just hover over that, you'll bring up a gray panel with a number of selections. When you're ready to ask a question, click on the Q&A chat box, and that'll open up the chat box in which you will post your question. Be sure when you enter your question to select all panelists as recipients so we can respond. Now within the chat box, just select Q&A, then select Ask a Question. Type your question, make sure all panelists are selected as the recipient, and then once you finish asking your question, there is a all Q&A section where you can see the questions and corresponding answers that others have asked. Okay, let's get started. Now, a lot of next-generation sequencing tools and analyses focus on short variations, small nucleotide variations, and indels. And certainly there are a number that are known to be causative of disease. These are all within, they're either point mutations or they correspond to small insertions and deletions that are less than 50 base pairs in length. But another major contributor to general variation and to disease variation are structural variations or large variations. And these can correspond to those types of alterations which don't alter the copy number. In other words, there may be inversions or balanced translocations which still remain in diploid with respect to any locus on the genome. Then there are copy number variations, which often correspond to insertions or deletions. And when I talk about insertions or deletions in the context of structural variation, these are on the order of greater than 100 base pairs. If you take a look at the figure on the right, and the top graph represents, let's say, the 4 blocks, consecutive blocks of DNA on the reference genome, a deletion would correspond to one of those stretches of DNA missing in the sequence sample. So here, block B. An insertion would correspond to a stretch of DNA added into the sequence, here corresponding to block X. An inversion is a little bit more complicated. 2 different stretches of DNA exchange their relative positions. So here, block C and B are found in opposing locations from where they are found in the reference genome. But rearrangements can be much more complex. Here we see that many of the regions are found in different relative positions next to each other, and this can even happen across chromosomes with translocation. Now, one of the points about the short variation versus large variation is that a lot of the more routine algorithms run for mutation detection will capture the smaller insertions and deletions, but the large insertions and deletions that are part of structural variation require dedicated tools for their detection. Now, what is the significance of structural variation in disease? I'll just say even within normal variation, structural variation from individual to individual corresponds to, at a base pair level, more than 100-fold difference when compared to, for instance, the SNV differences found between individuals. But even within disease, we find there's a significant contribution. Foundation Medicine has a panel for cancer diagnostics. They look at, in their first iteration of their panel, for which they published a validation paper, they looked at 287 genes and across 2,200 different patients. And what was interesting there was that this was their first panel, and while they looked at mostly exons for the 287 genes, there were a subset of about 19 genes where they sequenced 47 introns as well. So some limited intronic sequence where there were known to be a significant percentage of rearrangements. And this spans many different cancers, so it's also interesting to look at structural variation in that regard. And what they found was that 41% of the mutations detected either corresponded to gene amplifications, gene deletions, or rearrangements. So even when they were not looking at the whole genome, there was a significant contribution by structural variation. In a disorder with a significant heritable component, one good example is autism spectrum disorder. And this has got a lot of, has undergone a lot of investigation recently, not only by next generation sequencing methods, but also utilizing array CGH and SNP arrays designed to detect copy number variation. And just highlighting two of those studies, Marshall and Scher, for instance, found that unbalanced genomic alterations or copy number variation that are likely pathogenic occurred in up to 10% of autism spectrum disorder cases they examined. When Sabat et al. Looked at cases of sporadic autism spectrum disorder, where it did not run in families, they found that spontaneous CNVs were found at a rate of 10%. In other words, 10-fold higher than was found in controls. So here, too, there's a significant contribution by structural variation. Monogenic disorders can be a little bit more difficult to get an idea of in terms of the contribution by structural variation, but I think Spearfield et al. Did a reasonable assessment here when they looked at 91 strains of mice which represented models of human disease. And when they performed exome analysis, they were able to attribute the likely pathogenic basis for the disease to short variations, SNPs or indels, in 45% of the cases. And then what they did was they effectively reflexed for the unsolved cases to whole genome analysis. And in 17% of the remaining cases, they were able to identify a structural variation that was the likely cause of the disease. In other words, 8% of the total. So again, getting a good idea here that even in monogenic disorders, structural variation is likely to contribute to disease a significant proportion of the time. Now, at Bina, we have tools for both secondary and tertiary analysis. And in the secondary analysis tool, Bina, or rather module, Bina RAVE, we have both tools for calling SNPs and indels as well as structural variants. And we're going to feature in this talk the discussion of MetaSV. So on top of those individual tools for structural variation, we also perform an integrative or ensemble analysis. I'm going to hand over the presentation now to my colleague Margoob, who will go into a little bit more detail about each of those tools as well as the MetaSV tool.
All right, so thanks, Anu, for the great introduction. I'll go into detail about the methods that we use at Bina for detecting structural variations with high accuracy. So this is a more detailed figure showing the various kinds of structural variations. So for each kind of variation, on the top bar we show the reference genome, and on the bottom bar we show the donor genome. So as you can see, in case of deletion, the middle light blue bar region gets deleted and removed from the genome, so you get a removed sequence. Similarly, for insertion, we have the red sequence being added to the reference genome. And then you have duplication where a copy of a region of the genome is added and at the same location, so it's tandem. Or a copy of this region of the genome could be made somewhere else, which is called interspersed duplication. And then you have more complex SVs, like region of the genome gets inverted, or it just gets moved to a different region of the genome, and that's called translocation. So why is SV detection difficult? The problem is that with the current NGS reads, which are generally much shorter than SVs. So, for example, recently 100 base pair reads are very common read size for current NGS technology, and SVs can be of the order of even like a couple million base pairs. So which makes it difficult to detect SVs, and that's why we have tools to detect small variations and then SVs. And there are different methods to detect SVs. For example, read pair, split read, junction mapping, read depth, and assembly. And they have the strengths and weaknesses, and the accuracy that you obtain from a specific method can vary a lot depending on the size of the SV, the region of the genome, or the kind of SV. So this slide shows in more detail what the different methods involve. So for example, we have paired mapping, which looks at variations in the pair and mapping as evidence of an SV. So for example, if we have a abnormally large insert size, that's taken as an evidence of a deletion at that particular region, or an abnormally small insert size is taken as evidence for an insertion. Similarly, you have split read, which looks at individual reads and checks if you can align the read in a split manner on the genome in a very good way, and that's taken as an evidence of an SV. Or you have read depth, which looks at for variations in coverage in the regions of the genome. So if we have high coverage, abnormally high coverage, that is taken as an evidence of duplication, and an abnormally low coverage is as an evidence of deletion. And then we have junction mapping, which looks at a very well-developed database of known SVs, insertion or deletion, or any kind of SV, and then tries to map badly mapped reads or unmapped reads to those known SVs, and if those reads end up mapping very well to those known SV locations, then that's taken as evidence for the SVs. And then finally, we can simply take the reads in a specific region and assemble them completely to get the complete sequence of the SV in that region. So this slide quantifies in a better way how the different methods and tools vary. So we looked at very popular recent tools, Spindle, BreakSec2, Lumpy, which is very commonly used, BreakDancer, Delhi, Siminator, and Mind the Gap. And we use simulation to get a better idea of accuracy, because that gives you both precision and sensitivity. And we classified the SVs into different size bins. So we have 100 base pair, 200 base pairs, 200 base pairs, 400 base pairs, and so on. And we can see that as the size varies, the accuracy for each tool actually varies, and it seems to go up and down depending on the size of the SV. And each different tool has their set of sizes where it performs very well. And I also want to mention that these different tools, they actually have different breakpoint accuracies. So for example, Spindle can resolve SVs to single base pair resolution, while BreakDancer cannot, so it will give you very fuzzy breakpoints. So it's not just detecting SVs, it's also detecting SVs with good breakpoint resolution. So that is very simple, because each tool has different strengths and different weaknesses. So if you try to merge the tools in a smart way, then we can get much better accuracy. And the thing is that we want to make sure that the different tools that we use different methods, which are in a way complementary. So like we don't duplicate the work by using multiple tools using the same method. So if we use different complementary methods and then merge in a very smart way from those methods, then we can get a much better sensitivity and also precision. And there has been some prior work done on this integrative approaches. So we had SV merge and ISVP, and they had their own limitations, which we try to address in MetaSV. So here is a very simple example of why having multiple tools might be helpful and why a very simple merge may not be very helpful. So we have the green square, which represents the set of true positives. So if you make a call in the green box, you make a true call. If you make a call outside the green box, that's an incorrect call. So we have tool A, which makes the calls in the circle. So anything which is outside this green box is a false positive. So let's add another tool to the mix, tool B, which makes some calls which is the same as tool A, which is intersection, and some calls which is outside. So you end up covering more area in the green box, but you also cover more area outside the green box. So you also increase the number of false positives. So you increase both sensitivity but you lose precision. Then tool C covers even more uncovered area, but also adds more false positives, and then you have tool D. So we do see that if we add more and more tools, we do get higher sensitivity, but we also end up losing precision. And if you just take, let's say if you take the intersection of all the 4 tools, right, which is the inner, this area, you become very sensitive and practically, no false positives, but you also lose a lot of sensitivity. So what we have seen is that if it, so this just summarizes, if you use just the intersection of the 4 tools, you have lower sensitivity but high precision. If you try to use a more relaxed merging, so you say, okay, if a particular SV is called by at least 2 tools, then we report that SV, then you end up doing a better job in sensitivity and very few false positives. So overall, you get a very good accuracy. This slide shows a more quantified assessment of why integration is good. So this is from a recent paper. It was published 3 years ago. And the table shows that if you try to report calls which are made by multiple tools, we see a much better call quality. So if we use simply merge the calls made by all the tools, you end up having a very low precision. And once you are more stringent in terms of merging, let's say at least 2 methods to improve the accuracy in terms of sensitivity, and then if you, in terms of precision, and let's say if you want to merge at least 3 tools, then it gives you even more precision. So the solution to the problem that you mentioned is MetaSV, method-aware SV merging, and this was a paper that we published recently in Bioinformatics. So here is the MetaSV workflow. So what it does is that we run the alignments, the BAM files, through different tools using different methods. So in this case, the traditional MetaSV, we have 4 tools, each tool using a different approach for detecting SVs. So Brexic, which uses junction mapping, RegAnswer, which uses read analysis, Spindle, which uses split read, Simulator, which uses read depth. So we run these tools visually, and then we convert them to VCS just to have a unified format across all these tools. And then we try to remove duplicates among the calls with each tool, because sometimes because of errors and all that, we might end up having calls which are very close to each other. So we just try to resolve those duplicates first. So that's called the intra-tool merging. And then we do the actual, the core merge. So we merge calls which are common across multiple methods. And then if we see, okay, if a particular SV was called by at least 2 tools, we call them high confidence SVs. And if it's only 1 tool, we call them low confidence SVs. Now, we mentioned earlier that even though SV might be called by a particular tool and it might be correct, the breakpoint resolution may not be very precise. So we actually do local assembly to assemble the sequence for the SV, and then we align that assembled sequence against the reference genome to get precise breakpoints. Once we have the precise breakpoints, we genotype the SV, figure out whether it's heterozygous or homozygous, and then finally we report all the calls in VCF format, which is a very, very standard format for reporting variants. So what another thing I would like to point out is that, generally SV tools, deletions are the most easiest SV to detect. And we notice that most of the popular tools actually, they don't do a good job in detecting insertions. So the issue is that if none of those detect insertions with good enough insertions, we don't really, even after merging, we won't have enough insertions to report, and thus giving you low accuracy. So we added a software analysis to MetaSV to enhance insertion detection. And once we combine the software analysis with assembly to get very good and accurate insertion detection. So just to, I just want to clarify. So although we support assembly and figure out breakpoint more precisely, there's also an added cost of doing this. And it can take actually hours, if not days, to do the software analysis and assembly of SV breakpoints. So for the current version of RAVE, we don't really support assembly. We just do the merging across different tools. We do plan to support assembly once we have also optimized the assembly component of MetaSV. Right, so we now move on to the actual results, which compare the accuracy across different callers. So before I start on the accuracy, I would like to mention that our setup for assessing these different tools. So we used 2 kinds of assessments. One is using simulation, and second is using real data. So for simulation, we used the Watson framework, which was developed at Bina. And in the Watson framework, we did a simulation of 50x coverage, which is a typical coverage for a whole genome. And we used error profiles from the Illumina Platinum Genomes project. And the reads were paired and 100 base pair length. And we are trying to simulate the variants for the NA12878, which is a sample which is a very popular and well-studied sample in NGS variant calling. In order to assess both the breakpoint resolution and SV calling, we actually used a reciprocal overlap of 90% instead of the standard 50%. So we say SV is detected by a particular tool if the true SV and the found SV, they both overlap 90% with each other in terms of their length. So we're trying to penalize tools which don't report SVs with good breakpoint resolution. So we can see that again, different tools have different performance based on the size of the SV. And MetaSV, which is the top blue line, has the most stable performance. In fact, it almost never drops once you go beyond 200 base pairs. And it's not only the most sensitive, it's also almost the most, it's actually the most precise among all these four tools, all these tools. And F1 score, which is the harmonic mean of sensitivity assessment, is used to capture the overall accuracy. And finally, for real data, because for real data assessment, we don't really have the complete exact set of SVs. We can only measure sensitivity. And for sensitivity, we used the high confidence deletions for the NM12X78 sample from complete genomics. And with that, actually, we end up getting 90.2% sensitivity. So that means we call 90.2% of the calls made by complete genomics. And given that this is a different technology, I mean, we're trying to compare calls made on the Ilona Platinum sample against the complete genomics calls. A 90.2% sensitivity is a very good accuracy. So this was the accuracy for deletions. Now we move on to insertions. And as you can see, only Breakdancer and Mind the Gap and Pindle actually detect insertions, like more than 0 insertions. And except for Pindle, which ends up, which detects a lot of insertions, other tools detect very few insertions. But the problem with Pindle is that even though its sensitivity is reasonably high, it is very imprecise. That makes a lot of false positives, which ends up giving it a low F1 score. And if you look at MetaSV, it has the best performance by far on insertion detection. It's both sensitive and also very precise. So what other kinds of SVs can MetaSV detect? It's not the insertion and deletion. We can also detect inversions, duplications. We're currently working on adding support for translocations. And we're also working on improving the soft clip analysis to also incorporate other kinds of SVs. So I'll hand over to Anup to discuss more details of MetaSV on RAVE products.
Thank you, Margot. So as we mentioned, we have both modules for secondary and tertiary analysis, and it's the Bina RAVE module that both detects short variations, SNPs or SNVs and indels, as well as structural variations, presenting the results in the VCF format. Where in Bina RAVE is it found? Let's actually log into RAVE. Just a moment. All right, I'm in RAVE right now, and select Run Jobs, and then DNA analysis, DNA whole genome calling, and the first few steps involve specifying some metadata describing your sample, selecting reference or resource files, and specifying your output directory. Decide what quality metrics reporting you would like. You have a choice of multiple, you have a choice of BWA mem for alignment, and then GATK haplotype caller or GATK haplotype caller GVCS if you're going to be following up with a multi-sample analysis. Here's the section for selecting your structural variation tool. So as Margot mentioned, we've got Breakdancer, BreakSeq2, CNVnator, and Pindle, all as available options. As long as you select one of these, then in addition to the tool that you select, MetaSV will also be run in the back end. When you take a look in the View Job section of RAVE, then you'll find under the Status Monitoring a section that indicates the compute that was dedicated to MetaSV calling. Under the Browse Output tab, you can browse to find the folder corresponding to MetaSV, as well one for each of the individual structural variation callers, and from there access the corresponding BCS file corresponding to the results from each of those analyses. So now we're ready for our Q&A. If you're interested in trying the SV tools and the MetaSV tool, please go to our website and click to try the Take It for a Test Drive. We also have a research grant program for academics. And so if you have a specific project in mind, but you have not committed to the specific analysis tools or are considering different ones, and you think that structural variations would be very important to include, I would encourage you to apply for our research grant program. Just email info@bina.com and we'll provide you with more details of how to get access to both Bina RAVE, but also Bina AIM for the tertiary analysis. And actually, you can also email me directly for information, anoop@bina.com, to find more details about this program. Okay. All right, let's begin the question and answer session. Just as a reminder, the green scroll bar, if you hover over that, that'll allow you to select a Q&A button, which opens up a chat box. In the chat box, go ahead and enter your question, and be sure to select all panelists to receive the question so that we can all see the question and answer appropriately. In the chat box, you'll find that there's the option to click Q&A, ask a question, go ahead and enter your question, make sure all panelists are selected as recipients, and then click the Send button. All right, and then if you want to actually see questions that others are answering and the corresponding answers, you can go back and toggle to the All Q&A. All right. Let's get started with the Q&A.
Okay.
So our first question, what happens when only one tool is selected on the RAVE UI? Why don't you need at least two tools for the MetaSV to run in the back end?
So that's a good question. So basically, because I mentioned earlier that even if you use a single tool, it might still make duplicate SV calls, like calls which are roughly at the same location, so and reported as distinct SVs. So first of all, MetaSV will remove those duplicates just to make sure that they are less false positives. And secondly, because we don't really run assembly on the Bina RAVE product, so what we do is that although we don't run assembly, we still run HyperTalk caller from GATK to call small variants. So we actually use the HyperTalk caller as a proxy for assembly, So we try to see, okay, if a call was made by that particular tool and also by GTK Hypertalk caller, then it's reported as a high confidence SV.
Okay, the next question. What does it mean for MetaSV to run in the back end?
So what it means is that so once you have enabled, like selected one or more SV detection tools on the run jobs page, MetaSVs always run automatically by default. Because we don't really enable assembly, therefore the overhead of running MetaSV is like only a couple of minutes, and so there's no harm in reporting high confidence variants.
Next question. What is the overhead of running MetaSV?
So as I mentioned earlier, it just takes a couple of minutes to run if you don't enable assembly. If you enable assembly, it can take hours or days, depending on how many nodes you have.
Okay. The next question, how does MetaSV perform on DNA from FFPE samples compared to fresh?
Well, we haven't had a chance to assess that particular, those kind of samples, and we are working on actually doing more assessment of MetaSV on different kinds of samples. Yeah, so another question is that, have you done any joint SV calling using MetaSV? So MetaSV unfortunately doesn't do any joint calling, it works only for single samples. So those single samples have to be called using different SV calling tools. So question from Ms. Carolyn Perman. If I understood correctly, the BinaRAVE platform does not support local assembly at the moment. How does that affect the results for the customer? Well, for deletions, we don't expect assembly to improve things much in terms of accuracy, but yeah, for insertions, because the impact of assembly is very high, the customer would not see that many insertions from MetaSV. We do, however, report, because we report both high confidence and low confidence calls, so we report all the insertion reported by all the tools. It just ends up like being not that many high confidence insertions. And we are working on actually improving MetaSV in terms of performance, and once that is done, we will actually incorporate that onto the BinaRAVE platform. Question from Rashesh Sangvi. When you mention duplication events or deletions considering the coverage, how do you determine the normal? So we actually, because we only use simulation, we know exactly what the normal is. So we directly use that for the assessing the true or false positives. Question from Horacio, does Bina run normally in Linux, and which flavors have we tested on? So the Bina platform actually is based on Linux, and we have tested MetaSV on Linux, but because it comes packaged as a Python package, as long as, you can run Python with the required dependency of the tools, it should be possible to run. But again, we not really tested it on other platforms. So question from Palak Sheikh. What is the largest size deletion, insertion, inversion, and translocation that MetaSV can confidently call? So, okay, so let me first discuss deletions, inversions, and translocations first. So MetaSV doesn't really detect translocations. We plan to add support for translocations, although individual tools can actually call translocations because they're slightly more complicated than other kinds of SVs. Now deletions and inversions, there's no limit on the size. It just depends on like whether the individual tools can make those large calls. And then for the insertions, well, we can detect both long and small insertions, but the problem is that we cannot really assemble the long insertions completely. So if MetaSV detects a long insertion call, it might just tell you, okay, there is a high confidence long insertion at this location, but it would not know what the length of the insertion is because it just cannot assemble that long insertions with the short reads. Yeah. So what does the output of MetaSV look like? So it's basically a VCF variant entry, but we also annotate the individual variant with what tools call that variant, with what values, whether it's a high confidence or low confidence variant, and what is the genotype for that variant. So it looks like pretty much a standard variant entry in a VCF file, but we also add details about what kind of tools made that particular call. Jahangir Sheikh, did you play with the number of tools as a function of performance to come to a conclusion that if a SV is detected by 2 tools, it is sufficient to provide high accuracy? Okay, so we didn't really investigate like the different combinations. If I understand correctly, so we had 4 tools, and if you look at the different possible combinations, there could be 15 combinations to try out. We only looked at running all the 4 tools, and we saw that basically, adding more tools is good because you get better sensitivity. And we saw that for deletion, the precision was really high. So it is always beneficial to have more and more tools, especially if they use different methods. Question from Rashesh Sangvi. Have you tested with long reads? No, we have not tested MetaSV with long reads. We have actually also, however, investigated MetaSV with 250 base pair reads, so that seemed to work. If you look at our paper in the supplementary, we do mention the MetaSV accuracy assessment with 250 base pair end reads. So question from Ramzi Tamani, does MetaSV support schedulers? So as of now, MetaSV is designed to only use multiple cores on a single node. Once it's integrated onto the Bina RAVE product, it actually will be able to run on multiple nodes. But as of now, the Python script will parallelize only across a single node. Question from Paximadi, how do you handle the faulty results? Could they hide SNPs? So we don't really look at SNPs as of now, but that's a good question because a couple of false positives could be removed by examining SNPs in those SV regions.
All right, thank you. That concludes our webinar. If there's a question somebody posed that we haven't answered yet, we'll follow up with you individually by email. And we'll also send you a link to make a recording of this webinar available. Thank you so much for attending.
Thank you.